Hey, this is Rochelle, your microbiology tutor. Are you struggling to get A's on your exams? Are you struggling during your study time? Let's start right there so that you can get those A's in your microbiology exams. Well, today we're going to go over microbiology questions. We're going to break them down so you'll know what they look like and how you can answer them on your exams and get those A's that you want. Let's get into the video. Question number 1. Which substance in the following reaction is being reduced? Pay close attention to your questions. So, what is being reduced? Remember, it's not saying that what is reduced, but what's being reduced. And you also need to know what is reduction. Well, reduction is the gain of electrons. That's reduction. And when you see a reduction, there's always oxidation. And oxidation is the loss of electrons. Right, now that you know that, we're going to go to our reaction. We have acetaldehyde plus NADH plus H equals, or hydrogen, doesn't equal, gives us a reaction resulting in our ethanol and NAD plus. Now let's look at the first part of this reaction. We have acetaldehyde plus NADH plus hydrogen. The H in NAD, H and the hydrogen atom have protons, a proton and an electron. The electrons are going to go to or be added or gained by acetaldehyde. So we're going to have our electrons for our oxygen, and then our hydrogen will be added, giving us ethanol plus NAD plus. Now in our reaction, which one is gaining electrons? Well, it's our molecule acetaldehyde. So the answer is A. That's being reduced. And NADH is being oxidized. Question number 2. Which of the following reactions produces the most molecules of ATP during aerobic metabolism? So we want to know what produces the most molecules of ATP during aerobic metabolism. A. Glucose to glucose 6-phosphate. Well, this occurs in glycolysis, the breakdown of glucose. And in this reaction, ATP is actually being used. So ATP is used. That is a P. B. Phosphoenol pyruvic acid to pyruvic acid. This also occurs in glycolysis, the breakdown of glucose, and 2 ATPs are produced. C. Glucose to pyruvic acid. Now here, 2 ATPs are produced. And this is glycolysis. D. Acetyl-CoA to glucose, acetyl-CoA to carbon dioxide and water. We're going to come back to that one. E. Succinic acid and fumaric acid. Now in this reaction occurs in the Krebs cycle. The Krebs cycle. And in this reaction, FAD is oxidized. to give FADH2, and that one is reduced. So we do not see any ATP being produced. So 0 ATP. We're going to go back to D, and I will put D here. So with D, we have acetyl-CoA, which occurs between the pyruvic acid and our end product of carbon dioxide and some ATP. So we know that ATP is produced in this process, and we're going to say 1 ATP per pyruvic acid. Now we have acetyl-CoA to give carbon dioxide plus water. Now water is not produced in the Krebs cycle, however, it is produced in the electron transport chain. So we're going to go a little bit further, and we see that in the electron transport chain that 34 molecules of ATP are produced. Now in prokaryotes, we have 34 plus 4 ATPs to give a total of 38 produced. And in eukaryotes, we have 34 produced plus 2 ATPs to give 36 total ATPs produced. So which one, A through E, would give us the most ATPs? Well, the answer is D. Question number 3. Which of the following processes does not generate ATP? A. Photophosphorylation. It is the use of energy of the sun to produce ATP from ADD. So we have ADD to give ATP using energy of the sun. B. The Calvin-Benson cycle is a chemical reaction in chloroplasts during photosynthesis. So it is using ATP. C. Oxidative phosphorylation. It is the series of oxidation reactions leading to ATP synthesis. So it is creating ATP. So it's a production of ATP. D. Substrate level phosphorylation. ATP is produced in this one. So the phosphate groups of substrates leave to bind to ADP or GPP. So there's a production of ATP because the substrate or phosphate groups of substrates leave to produce ATP. And they bind to ADD, or that is a D, or GPP. Now all of the above generate ATP. Well, the answer cannot be E. We have A that produces ATP. We have C that produces ATP. And we have D that produces ATP, which would leave us B of being the answer because it uses ATP. All right, let's go to question number 4. Let's keep moving. Question number 4. Which of the following compounds has the greatest amount of energy for a cell? A. Carbon dioxide. Well, that is released. B. ATP is released and used, or produced and used. So we're going to do produced and used. Glucose is a molecule that is oxidized. D, the oxygen, the molecular oxygen, it's produced and used as an electron. Well, it's used as an electron acceptor, final acceptor. It's electron, final acceptor. And E, lactic acid. It is the end product of fermentation. Oops, that's supposed to be a P. Product of fermentation for some bacteria. All right, going back to the question. Which of the following compounds has the greatest amount of energy for a cell? What can give the cell energy? Well, the answer is C. Because of glucose entering in the reaction, ATP is produced, giving ATP, giving the energy. And like I said in the previous question, that prokaryotes and eukaryotes produce ATP, but specifically we have 38 for prokaryotes, and because they have an extra 4 from glycolysis, and we have 2 extra for eukaryotes, which gives 36. So that gives, the glucose gives us energy, and prokaryotes energy. Question number 5. Which of the following is the best definition of the Krebs cycle? In order to answer this question, you need to look at the question a little bit deeper. So we have best, we want the best definition of the Krebs cycle. Now let's focus on cycle. What is the definition of a cycle? Well, a cycle is a series of events that are regularly repeated in the same order. Now, in our options, where do you see a series of events? Well, it's not A, so we can just put a no there, or just cross it out. The way cells produce carbon dioxide, no. A series of chemical reactions, yes, we see that. D, a method of producing ATP, no, we don't see that. And E, a series of chemical reactions. So we see a series of chemical reactions in C and E. Now we need to go in deeper to know what the best definition is. So C, a series of chemical reactions in which NADH is produced from oxidation of pyruvic acid, which is true, and a series of chemical reactions in which ATP is produced from oxidation of pyruvic acid. Well, there is a small amount of ATP that is produced. We are going to go with the answer C. That is the best definition of the Krebs cycle. Question number 6. Which of the following is the best definition of respiration? A sequence of carrier molecules with O2 as the final electron acceptor. B, a sequence of carrier molecules with an inorganic molecule as the final electron acceptor. C, a method of generating ATP. D, the complete oxidation of glucose to CO2 and H2O. And E, a series of reactions in which pyruvic acid is oxidized to CO2 and H2O. In respiration, we see glycolysis, in the Krebs cycle, and then the electron transport chain, where we see a bulk of our ATP being produced. Now that we know this, let's go back to our options. Now, we know glycolysis, Krebs cycle, and ETP. That's a series or a sequence of events. So we can go back to our options and we can eliminate C and we can eliminate D, which leaves us with A, B, and E. A sequence of carrier molecules with O2 as the final electron acceptor. We do see that, but is it the best definition? And is it the definition that we see from reading our textbook? No, so we're going to cancel that one out. E, a series of reactions in which pyruvic acid is oxidized to CO2 and O2. Well, glucose. Glucose is oxidized. Glucose is oxidized, and we see that CO2 is produced or released from the Krebs cycle, and O2 is produced in the electron transport chain. So we are going to cancel E. So what we're left with is B. So the answer is B. That is the best answer. Inorganic molecules would have to come in or found outside of the cell and come in to be the final electron acceptor. Now we have our last few questions. Use the following choices to answer questions 7 through 10. A. Escherichia coli growing in glucose broth at 35 degrees Celsius with O2 for 5 days. B. Escherichia coli growing in glucose broth at 35 degrees Celsius without O2 for 5 days. C. Both A and B. D. Neither A nor B. Now let's break this down. We have 7. Which culture produces the most lactic acid? Well, we see that we know that lactic acid is produced during fermentation. And fermentation is anaerobic. That's without oxygen. Oops. So the answer is B, without oxygen. 8. Which culture produces the most ATP? So E. coli growing in glucose broth at 35 degrees Celsius with oxygen or O2 for 5 days. Well, with oxygen or with O2, we have with O2 aerobic respiration. And in aerobic respiration, we have a ton of ATP being produced. So the answer is A. 9. Which culture uses NAD+? Well, both of them do, because it goes through glycolysis, It goes through the Krebs cycle and the electron transport chain, where the cultures use NAD+. So the answer is C. 10. Which culture uses the most glucose? Well, the answer is B. Why is it B? Because in fermentation, the process is very short and the turnaround is quick. So then they'll be able to produce more or use more glucose since the process is short, the turnaround is short.